And I want to introduce Hannah, who will be our second speaker today. Hannah finished her work at University of Virginia, and who is a fantastic and extremely productive graduate student. And I would encourage you, any students in the audience, to ask her about what's her success in doing such a fantastic graduate studies. And then she will be talking about as well, also microglia and relationship with microglia and Alzheimer's disease. And Hannah, please take away.
Thank you so much. Let me go ahead and share my screen. Can everyone see this okay?
It's perfect. Thank you.
Okay, awesome. Yeah, thank you so much for the introduction. So today I'm going to be talking to you all about the protective microglial signaling that can occur in neurodegenerative diseases. As mentioned, I did this work in the Brain Immunology and Glia Center at the University of Virginia, which was an amazing place to train for neuroimmunology. So if you're looking for a home there, right now I'm currently pursuing a postdoc at Stanford, but you know, UVA is always my home. And the paper that I'm going to be talking to you about today is, this paper published in Cell a couple months ago, titled SYK Coordinates Neuroprotective Microglial Responses in Neurodegenerative Disease. And we did this work in collaboration with Marco Colonna's lab at WashU, had a very similar paper, which you would have seen in the same issue. But to begin, just an introduction on Alzheimer's disease. As you all know, it's very prevalent. About 1 in 9 people above the age of 65 are diagnosed with this disease. And functionally, it's shown to be described with cognitive decline, and the risk factors for this include age, genetics, and lifestyle. And the pathology for Alzheimer's disease is pretty well described. One of the first pathologies that shows up is amyloid beta. And this is a protein that can accumulate into plaques, which is a hallmark of disease. And then following amyloid beta accumulation, you can then get tau pathology, which can form neurofibrillary tangles that can contribute to neuronal loss. So that's going to contribute to cognitive and memory decline. And then also you get microglia expansion, also known as microgliosis, which I'll go into a little bit more detail later. But I always show this graph just to impress upon everyone the importance of amyloid beta in the disease, because this particular protein accumulation occurs sometimes 15 to 20 years in advance of cognitive impairment before you have any sort of memory decline. So scientists for many decades have really been focusing on this pathology in hopes to curb the progression of the disease. Because unfortunately, once you start to get tau accumulation, you already have neuronal loss that's very difficult to reverse. So it's thought that by targeting amyloid beta, we can really stop the snowball effect of this disease. But of course, my papers focus on microglia, and who are they? So microglia are these resident innate immune cells of the brain, and in homeostasis, they have a lot of functions, including maintaining the blood-brain barrier, so making sure nothing gets in that's not supposed to get into the brain. They're also important for pruning synapses and generally surveilling the brain for anything that could be awry. However, in Alzheimer's disease, they have a slight shift in their function, one of which is that they start to eat the pathology that can accumulate in the brain that is neurotoxic, including amyloid beta, and they'll do this through a process of phagocytosis. However, microglia have many different functions outside of eating things, so outside of phagocytosis, and this is what I love to focus on during my PhD, because I think that microglial biology is very fascinating. But one of them is that microglia are great proliferators. So in the event of any kind of disease onset or pathology, they're going to proliferate in number. They also produce many inflammatory cytokines. They can be recruited to plaques, things such as amyloid beta plaques or damaged neurons. And in the case of amyloid beta, they'll form this nice barrier that surrounds amyloid beta. So this compacts the plaque as well as protects surrounding neurons from the neurotoxicity of it. And in addition, they can activate. So this activation can happen morphologically. So at rest, they're more ramified. And as they become activated, so they have some sort of triggers and disease, they'll become more amoeboid. However, in addition to morphology, they can also activate transcriptionally, in which they become disease-associated microglia. And this happens when they are in the environment of things such as amyloid beta, in which they downregulate their homeostatic markers and upregulate things such as TREM2, which is a really well-described microglial receptor in the context of AD. And these receptors, including TREM2, are important for recognizing what's going on in the brain environment, including maybe binding amyloid beta itself or other ligands associated with Alzheimer's disease pathology, which could also include things like lipids from the damaged neurons. And there have been several microglial receptors that have been well described in the field, including TREM2, as I mentioned, but ones that are also coming to the forefront include SYK7A, CD33, and CD22. And they all have variable ligands. And the one thing that we do know about them is their ligands and their function in Alzheimer's disease, whether or not they're, protective or non-protective. But what we didn't know until this past year is a lot of the downstream signaling that occurs of these receptors. And a lot of it was assumed from the peripheral counterparts of microglia, macrophages. So to fill in the blanks, we were very curious about the downstream signaling of these particular receptors in hopes of maybe identifying a new therapeutic target. But at first, we really wanted to see what's going on at a greater level. And one of the things that a lot of these receptors are hitting is this one molecule called spleen tyrosine kinase, which is pretty far upstream, a caveat of our study, but we were very curious about how SYK could be regulating microglial response in the context of neurodegenerative disease. And in order to do this, we did a selective deletion of SYK in microglia and an AD model of mouse, which is called the 5xFAD mice. And these mice have amyloid beta accumulation beginning at 1.5 months of age and memory decline beginning at 4 months of age. And basically what we do is we selectively delete SYK using a tamoxifen diet and a CX3CR1 ER22 Cree mouse line. And basically they receive tamoxifen at weaning, so 3 weeks before you actually get amyloid beta accumulation. They're taken off 2 weeks later, and this allows for the peripheral CX3CR1 expressing cells to turn over and regain SYK expression while leaving the long-lived microglia SYK deficient. And as you'll see in the study, I'll refer to our littermate controls as just 5xFAD, and then SYK delta MG is just our change in microglia, so SYK deficient microglia on the 5xFAD background. And we take these mice at 4 months and run behavior, and at 5 months we harvest the brain to look at pathology. And the first thing that we notice in these mice is that SYK deficient 5xFAD mice have a higher level of amyloid beta in the cortex, hippocampus, and the thalamus, all regions that are characteristic of amyloid beta accumulation. And looking at the neurons, we saw an accumulation of dystrophic neurites, as labeled here in APP, surrounding a THIO-S or pink positive amyloid beta plaque. And this is usually indicative of just them being very unhealthy and on the verge of starting to begin the process of dying. We also saw an increase in hyperphosphorylated tau, as labeled here with AT8, surrounding our amyloid beta plaques in the 5xFAD SYK deficient mice, which, as I mentioned in my introduction, is kind of the following pathology that occurs after amyloid beta accumulation. As well as we saw an increase in dying neurons in the CA1 region of the hippocampus, which is a tunnel positive stain of these neuron positive neurons in the hippocampus in the SYK deficient 5xFAD mice. All in all, really suggesting a breakdown of this neuronal integrity and made us wonder whether or not there would be impaired memory in these SYK deficient 5xFAD mice as well. And in order to look at this, we use the Morris-Water maze, which I'm sure you all are familiar with, but I'll give a brief introduction. It's basically where we fill a pool with opaque water, and the mice have different signaling visual cues on the side of the pool, and they have a hidden platform, and they should be trying to find the platform to escape swimming. And as they learn, they should be decreasing the amount of time or latency to find the hidden platform. And as you can see, the SYK deficient 5xFAD mice spend more time looking for the platform. And on the 5th day, when we remove the platform, they spend less time in the target quadrant of the pool that the platform used to be located, all in all suggesting there's an impairment in spatial learning and memory that's exacerbated in these SYK deficient 5xFAD mice. However, as I mentioned before, we were deleting SYK before pathology accumulation. So we were very curious whether or not SYK was also regulating pathology accumulation and progression. So in order to do this, we deleted SYK later on. So we waited until 4 months to delete SYK after pathology has occurred, and then we harvested the brain at 7 months. And as you can see, a very similar trend occurs in the SYK deficient 5xFAD mice, in which they have more amyloid beta accumulation, which indicates that SYK is not only important for AD development, but also pathology progression. And what I've shown you so far is that the loss of SYK in microglia exacerbates AD-related phenotypes. So very broad level of this is what's going on with the pathology. But what actually happens to the microglia? Because that's what we're directly deleting SYK from. And we're really curious about how this could be affecting pathology itself with these cells. And the first thing that we notice is a striking decrease in the number of microglia in the SYK deficient 5XFAD mice. As you can see, they also form less of these plaque-associated barrier formation. So you no longer get these microglia really converging on the plaque in the SYK deficient mice. Therefore, this can really increase the toxicity of the plaques on the surrounding neurons. So we suggest here that SYK is critical for driving microglial recruitment to amyloid beta plaques in the context of AD. And the decrease in number led us to just look at proliferation markers. So we just use a Ki67 stain. And as you can see here, the SYK deficient 5XFAD microglia proliferate significantly less than their 5XFAD counterparts, which may suggest or may explain why we see a stark difference in number between the groups. Also suggesting that, SYK is important for driving microglial proliferation in the context of AD. And next, we wanted to look at activation. And as I mentioned before, microglia at rest are really ramified. And when they become activated, they become more amoeboid and lose these processes. And the way to study this, we just use a shoal analysis in which we draw concentric circles, 1 micron all the way to 50 microns out from the cell. And essentially what we do is we count intersections, so the number of these processes within each circle. So number of intersections is on this axis here. And as you can see, the SYK deficient 5XFAD microglia are much more ramified, therefore more homeostatic in nature in terms of morphology. So without SYK, it seems that these microglia aren't able to become fully activated morphologically. And I think it's an important time to step back and just, this is a really important control to have. We don't really see differences in microglial number or activation at rest. So without the amyloid beta or AD pathology push, there doesn't seem to be a difference in the microglia themselves in the non-5XFAD, SYK deficient and deficient mice. But to look at it a little bit with a better lens, we wanted to look at the transcriptional shift that might occur in these mice. And to note, we just use CD11b beads to sort for microglia, and then we submitted them for bulk RNA sequencing. And what you can notice is the non-5XFAD groups cluster very closely together on this PCA plot, suggesting that they're very similar. However, the 5XFAD microglia cluster very far away from our controls, suggesting a major transcriptional shift that occurs in the context of AD. But even more strikingly is the SYK 5XFAD mice microglia, they actually remain clustered with our controls, which suggests that SYK is critical for that massive transcriptional shift that you normally see in the AD environment. And in addition to the major transcriptional differences between these two groups, we most strikingly saw a decrease or an inability of these SYK deficient 5XFAD microglia to take on the disease-associated microglia transcriptional shift, both in stage 1 and even more strikingly in stage 2. All in all, suggesting that SYK is critical for driving microglial activation, both morphologically and transcriptionally in AD. However, as I mentioned before, phagocytosis is a very major component of and one of the major functions of microglia. And so we aim to test this in vivo using a very elegant method we adopted from Marco Colonna's lab, in which we inject our 5XFAD and SYK deficient 5XFAD mice with methoxyXO4, which is a dye that's going to cross the blood-brain barrier and label amyloid beta. We do this injection at 5 months, and then we wait 3 hours after injection, and we harvest the microglia from these mice, and then we analyze them by flow cytometry to see the amount of methoxyXO4 tagged amyloid beta that's been taken up in the microglia. And as you can see, the 5XFAD microglia, about 30% of them have taken up amyloid beta, whereas in the SYK deficient 5XFAD microglia, almost none of them have taken up A beta, suggesting a major phagocytic deficit in these SYK deficient microglia. Therefore, SYK is really important for driving microglial phagocytosis in the context of AD. However, a lot of what I just described is what's going on at this level, but we want to get a little bit further down in our signaling pathway so as to maybe get a more targetable part of the pathway, and also just to fully understand the role and what signaling pathway SYK is taking in the context of AD. And the first pathway that we chose to look at is the PI3K and AKT signaling pathway, because it's been well described to be affected in the TREM2 receptor. And the way that we did this is we also harvested 5XFAD and SYK deficient 5XFAD microglia, and we incubated them with an ELISA dot that's specific for phosphorylated AKT and its downstream phosphorylation targets. And as you can see, the SYK-deficient 5-XFAD microglia have reduced phosphorylation at the AKT site, as well as the only downstream phosphorylation target affected is a decrease in the phosphorylation of serine 9 and GSK3 beta. And the way that this works is normally AKT will be phosphorylated by SYK, and then AKT will phosphorylate GSK3 beta, the serine 9 site, which actually inhibits its activity. But what we think is happening here is that with the loss of SYK, you lose phosphorylation of AKT, you lose this inhibitory phosphorylation at serine 9, and this increases GSK3 beta activity, which has been associated with amyloid beta accumulation. So to get at this a little bit further and really close the link, the circuit of signaling and how this could relate to the increase in amyloid beta that we see in the SYK-deficient 5-XFAD mice, we then just did a very simple in vitro experiment using bone marrow-derived macrophages in which we have wild type and SYK-deficient BMDMs or macrophages. And we treat them with Cypher5E amyloid beta. So this Cypher5E tag on amyloid beta, it will fluoresce in the low pH of the phagolysosome. So when something's being phagocytosed. And you can see just at rest, the SYK-deficient BMDMs have reduced levels of amyloid beta uptake, so impaired phagocytosis, which mirrors what we see in the microglia in vivo. However, when we add a GSK3 beta inhibitor called tiglucib, you can see a rescue in the amount of amyloid beta that's taken up, so a rescue in the amount of amyloid beta cleared by these macrophages. Really closing the circuit and suggesting that this inhibition of GSK3 beta can rescue this SYK insufficiency, and ultimately decreasing the amount of amyloid beta. However, we just went downstream, and there's a lot more that we really need to do on that. But let's also go upstream. You know, one of the things that our collaborators did a great job on is looking at the role of TREM2 and SYK signaling. However, we were really curious about another receptor that's gone a little bit underdescribed in the field, but is a disease-associated microglia marker. And we hoped that through the activation of this receptor that has been shown to signal through SYK, if we activate it, we can actually enhance SYK signaling. So the opposite of what we've been doing when we've been selectively deleting it in microglia and seeing detrimental effects. If we add it back, can it actually be a positive or be beneficial in the context of AD pathology? And the way that we did this is we treated our mice with a beta-D-glucan, which is a ligand for this receptor, and we used one called pustulin. And essentially, we took our two mouse groups, and either side of the brain, they received vehicle or pustulin. And this acted as an internal control for each mouse. And 7 days after injection, we looked at their hippocampus that had received these injections. And you can see in the 5-XFAD mice, you see a reduced amyloid beta load on the side of the hippocampus that received the pustulin injection, suggesting that the activation of SLEC7A seems to be very protective in driving amyloid beta clearance. However, when you do the same experiment in SYK-deficient 5-XFAD mice, you no longer see this trend, suggesting that SYK is critical for that protective SLEC7A-induced clearance of amyloid beta by, we presume, microglia. And what I've shown you so far is that microglial SYK is protective in AD, but we were also very much wondering whether or not microglial SYK is relevant in other neurodegenerative diseases. And the one that we look to next is multiple sclerosis, which there's about 1 million cases in the US right now, and symptoms include impaired loss of vision, trouble thinking, muscle weakness. They're very varied. And the risk factors for this include age, sex, lifestyle, and infection exposure. Now, normally in homeostasis, our neurons are wrapped by this wonderful myelin sheath that allows for proper conduction and protection of the neuron. However, in the case of multiple sclerosis, you start to get demyelination, so this autoimmune attack on the myelin itself, leaving these neurons actually susceptible to neurodegeneration. And microglia also have varied functions in the context of MS, including proliferation of themselves, but also driving proliferation of oligodendrocyte precursor cells, or OPCs, which are the precursor cells for oligodendrocytes, which actually myelinate our neurons. So really important factor, not just signaling through to themselves, but also to other cells. And they also can produce growth factors for neurons. They can phagocytose and recruit to damage myelin basic protein that accumulates during disease. And they can also activate morphologically and transcriptionally. And in the lab, we've used well-characterized mouse models of MS. We used EAE and Cooperzone, which I'll go over in a little bit. And a lot of the EAE work was done by Dr. Elizabeth Ross, which was a postdoc in our lab at the time. And she did a really elegant experiment, which I will describe later in single cell sequencing, but also in her EAE experiment where she just induced EAE using MOG And she collected the spinal cord after assessing the clinical score for these mice. And the clinical score is just basically the hind limb weakness and paralysis in these mice that occur after EAE induction. And you can see that the SYK deficient mice have an increased clinical score, so increased weakness. And this is coupled with a decrease in the amount of myelination in the spinal cord as measured by Luxel fast blue in our SYK deficient mice. So an exacerbation of disease upon the loss of SYK. And when she took these microglia for single cell sequencing, we basically were able to find 6 clusters. And what I hope you can notice is that the SYK deficient microglia remain in the homeostatic hump and fail to go really into this DAM hump, which the SYK sufficient microglia seem to really be in this DAM or disease-associated microglia cluster over here. And if you look at the proportion, you can see a little bit better that the SYK deficient microglia, they're less DAMs, they're more homeostatic, and they're more in the CD36 cluster. And we named this cluster CD36 because it was one of the defining genes for this cluster that was very highly upregulated. And it's very curious because it's a scavenger receptor that recognizes amyloid beta and is independent of SYK. So we thought this could potentially be a compensatory mechanism that these 6 deficient microglia could be taking in order to respond to amyloid beta. But of course, a lot of work is needed in order to actually verify this. And so here we can say that SYK is critical for driving microglial activation, at least transcriptionally in the context of demyelination. But one of the caveats of EAE is that there's a lot of other immune cell types that are important for driving the disease. It's not just microglia. Of course, T cells are the main players here. So we wanted to shift to another model of demyelination in which microglia are the main cell type that's going to be responding, the main immune cell type. And we did this using the cuprazone diet, in which we get really nice demyelination in the brain. Specifically, we have a nice area of demyelination in the corpus callosum. And essentially what happens is we give them this diet for 5 weeks, and then we harvest their brains, or we put them back on normal diet and harvest them a week later to allow for remyelination. But an important control is that the SYK groups don't have resting differences in the amount of myelination in the corpus callosum. So that's not describing the differences that we will see. And what we first saw is that during demyelination and remyelination, SYK deficient mice had reduced numbers of microglia, which is very similar to what we saw in Alzheimer's disease, kind of mirrors what we see in this other neurodegenerative disease. We don't know whether or not this is due to proliferation and recruitment. That's still up in the air, but still it seems that SYK is regulating microglial numbers in demyelination. And even more strikingly is that there's an accumulation of disease of damaged myelin basic protein during demyelination and remyelination in these SYK deficient mice, suggesting that the SYK deficient microglia are not able to properly clear this damaged myelin that accumulates during demyelination. And we would suggest from this that SYK is critical for driving microglial phagocytosis during demyelination. But finally, I think what is really interesting, and this is something that Dr. Elizabeth Frost did, is she looked at the corpus callosum for 2 different cell types, OPCs and oligodendrocytes. And what she saw is that the SYK deficient mice had reduced numbers of OPCs, oligodendrocyte precursor cells, during demyelination and remyelination. And this culminated in there being less oligodendrocytes during remyelination, which are these myelinating cells, which can contribute to the fact that there seems to be a failure to remyelinate and an accumulation of this damaged myelin basic protein, ultimately suggesting that microglia, that SYK in microglia is not just important for driving differential microglial response, but also in driving other cellular response, including OPCs and oligodendrocytes. And what I hope that I can impress upon you here is that similarly to what we see in the Alzheimer's disease model, SYK seems to be critical for driving microglial response in the context of demyelination. And as a summary slide, SYK is critical for recognizing amyloid beta and damaged myelin ligands in the neurodegenerative environment. And SYK also drives the disease-associated microglia phenotype, the transcriptional shift that happens in microglia, which can be protective in clearing amyloid beta pathology. And without it, we have an exacerbation of disease due to the retention of these microglia in a homeostatic state. And with that, I'll take any questions. I want to give a huge shout out to my grad school PI, John Lukens, who I did all of this work with. And Of course, Dr. Elizabeth Frost, who did a lot of the EAE work, and Daniel Shapiro, who did a lot of the sequencing work as well. And yes, I'll take any questions.
If you have any question, please raise your hand or directly unmute yourself and shout out the question. And all right, let me see. We seem to have one question popping. I'm wondering if you are, I'm just directly read the question. I'm wondering if you are, you also look at B cells or monocytes levels in the presence or absence of SYK. I think a glioma model that pharmacological blockade of SYK alter as well as the level of those cells. And I guess in terms of the B cells and monocyte level.
Yeah, so I guess the question is maybe pertaining to the actual deletion of SYK in our model in other cell types outside of microglia, which could be a caveat. Is that correct? Kind of?
No.
Oh, sorry. Let me,
yeah, I
on,
wonder because, sorry, yeah, I wonder not only because of that, but also because, of course, in the other papers, I know it was like more pharmacological, so maybe it was also an effect of that. But the way the authors them, it was more related to the absence of, well, the blockade of SYK in microglia. Yeah. So I wonder if here it will see something different, but again, it's not my topic, so maybe those cells also don't play a role.
No, they definitely do express SYK. Many different cell types express SYK, and that is another caveat of actually targeting SYK in the context of AD. I mean, obviously, we couldn't just add, a SYK inhibitor or a SYK activator in the context of these because it's going to be hitting many different cell types, which can have a lot of effects. And SYK has been studied in the context of T and B cells in cancer as well. And so I think that it's really important to make that point, that there is expression in other cell types, and any sort of targeting of SYK itself or downstream targets will have to be specific to microglia in some way or another, or we have to take into account that there's other side effects due to the effect on other immune cells. But we didn't really look at B cells. We did look at T cells just to look to see if SYK expression went down when we did this particular model, because we wanted to make sure that we were just hitting microglia, and we didn't see a change in SYK expression in T cells in this particular model, just as a side note.
Thank you.
Mm-hmm.
Hannah, I just want to ask two questions that's really out of my sort of limited knowledge about Alzheimer's disease and microglia. And the first question is this surprising to you? The function of SYK seems to be very sort of specialized for information states of the brain or like in the AD condition, but not during the development. So sort of the question will be wondering like, what's the role of this signaling pathway? So is it just sitting there protecting, just anticipating any disease that sort of wanted to provide the protective function, or like what's your thought about this the function, the fundamental function of this signaling pathway? And then the second, sorry, please go ahead.
Oh yeah, so I think what you're referring to is the fact that we didn't really see changes in homeostasis in terms of the microglial activation, but when we give the push, so you also saw this in the demyelination models too, you didn't see a change in microglia number or activation in homeostasis, but it needs actually this neurodegenerative push, or these ligands that really seem to be driving this differential response. And so, SYK is expressed all the time, but I think that in terms of the downstream activation in which it's actually pushing, which are the effects that we're really seeing in driving differential microglial response in both contexts of neurodegenerative diseases, I think that is then driving or, kind of instructing these microglia to respond in a different way. And I think that it's probably ligand dependent. So in this case, we're just looking at myelin and we're just looking at amyloid beta specifically. But I think it'd be really interesting to look at different triggers to see how this can drive differential response, including like an injury model, for instance.
Fantastic. We have another question coming in from the chat. Great presentations. You were mentioned that SYK is involved in AD development as well as progression. Could you comment on whether SYK's involvement in phagocytosis is the mechanisms in both AD development and progression, or whether it could have different mechanisms in each stage of AD?
That's very interesting. We didn't really look at phagocytosis at a later stage in terms of the methoxy experiment. We did it earlier on. I would suspect that it's probably a similar pathway. There could be some shift at some point, but I think that there's, we still were giving them time for the disease progression to occur. So there were still higher levels of amyloid beta at that point, that the difference really could have been clearance of amyloid beta. But I think that that's a really great question, and we never looked at that in full. But I would imagine that because these microglia seem to fail to actually meet at the plaque, really form that barrier and actually ingest it at 5 months, I would imagine a very similar case is occurring at the 7 and 8 month mark.
Thank you so much. And please give another round of applause for Hannah and Julia for their fantastic presentations. And thank you for joining us today. We hope you all have a fantastic holidays ahead, and good luck with all the adventures in 2023.